Okay, so the name literally means, platy means flat. You've heard like the platypus, the duck-billed platypus in Australia, which has a flat mouth, like a duck beak. Platy means flat. Helminths is a Latin term used for worms. Specifically, it kind of refers to parasitic worms. So helminths would be like flat parasitic worms. Burmese, we're going to look at Tuesday, which is the round worms. So there's a whole bunch of little parasites that we're going to look at in these examples, right? So what we're getting into here is more common features with the rest of the animals that we know. So this includes the flatworms, the flukes, and the tapeworms. They're kind of the common names of these things. Here are kind of the 3 bullet points. So you'll notice that I did this last time, that I kind of wrote some like key features of this phylum that you have to know, right? On the last one, I had kind of some 3 bullet points of the Nigeria that were important. Here we go again. These have what's called triploblastic development. The jellyfish and hydra, remember, had diploblastic, which means 2 tissue layers. Jellyfish and a hydra had, anemones, had endoderm and ectoderm only. This group has endoderm, ectoderm, and a new tissue layer called mesoderm. This is what's called triploblastic. Triplo meaning 3. Blastic means tissue development. Triploblastic development. And specifically, we have endoderm plus mesoderm plus ectoderm. And the new tissue layer here that we're interested in is the mesoderm. And what we're going to see is mesoderm ends up taking over as the dominant tissue. So most of your body mass is your muscle and your internal organs, which are all mesodermally derived. Your epithelium, your integument, is ectoderm, and your intestinal lining, the lining of your intestine, just the inside lining, is endoderm. So throughout the rest of the semester, we're going to see is that mesoderm makes up the bulk of animal structure. So this new tissue probably resulted in animals being larger and more kind of durable, and certainly muscle movement is a key thing that comes up with mesoderm arrangement. So mesoderm is important. Bilateral symmetry. This is an important one. We knew that the jellyfish and the hydra anemones were radially symmetrical, meaning that they had a mouth and a non-mouth. Otherwise, they were kind of like a pizza or like a circle type body plan. These have bilateral symmetry, which means they have a left and a right side. Bi means 2, lateral means sides. So they have 2 sides. They have left side and a right side, but they only have 1 head, 1 tail, 1 top, 1 bottom. So what bilateral symmetry means is they have orientation where they have 2, you can only cut them in 1 plane. I'll show you a picture here. But this is caused by cephalization. And what that means is we have a head forming. So when an organism basically has an anterior end, as a result, everything else falls in place. The opposite of the anterior is posterior, and then you have a dorsal and a ventral. Each one of those only has 1, but you have 2 lateral sides. That's why it's called bilateral. The third one I'm not going to write down because it's kind of like a general description, but these are the 2 key things that you'll have to answer on the liabilities as to what the kind of the key steps of the development here. Okay, let's talk about this triploblastic development thing. What we have going on is a body plan. Let me go ahead and just draw like a generic flatworm here. So we have like a flatworm. This is kind of like a free-living flatworm, planarian here. have our what bilateral symmetry is. At one end of the organism, they developed eye spots. And these eye spots are pigment spots that are able to see, basically light and kind of some real primitive motion. Associated with those eye spots, they have developed neural ganglia, or head ganglia. So this end becomes kind of the sensory end, like for light, for vision, for sight, and also for touch and tactile orientation. So it's kind of like the decision-making center of the body. And as a result, it's the area of the body that kind of leads the way, right? It's kind of like making decisions and sort of leading the way. So thus, this is cephalization. So this is the anterior. The opposite of anterior is posterior. Now I'll explain why it's not called oral and aboral, because the mouth is nowhere near the anterior end or the posterior end, it's on the ventral side. And if we look at ventral, we're looking at a side view. This is like a top view of the animal. A side view of the animal would look something like this. Like if we were to look at it from the side, their mouth is actually a pharynx, like a vacuum hose that extends from the ventral side. So if we look at a side view of the animal, we have the eye spots are here, like this. So we have dorsal, we have ventral. Okay, so here's anterior orientation. There's one head, there's one tail, posterior. There's one dorsal, one ventral, and then we have lateral. This would be like left, and this would be the right lateral. So because there's two laterals, that's why it's called bilateral symmetry, right? Or I've already mentioned that, but I'll say it again, because that's gonna be a test question, right? Not this. Okay, so that's what the orientation of symmetry means. Triploblastic development, if we were to take a cross section, let me show you how the gut is formed. The gut is formed by a gut pocket that has a gastrovascular cavity. So this pharynx here, this is a muscular mouth opening. It's kind of like a vacuum hose. The pharynx is how it reaches out to feed, is by this kind of vacuum hose nozzle that sucks up food. And the pharynx opens up into a gut pocket. A lot like what the Cnidaria had. So we have this gastrovascular cavity. And yeah, on the test, you're going to have to write out gastrovascular cavity. That's like just one of those things. You just have to know it. So you can't just say gut. Gastrovascular cavity, okay? And it's because it's a gut and a circulatory cavity, where when the food's brought in, it's digested by enzymes and then it's distributed throughout the body by that pocket. In the dorsal view, what you see basically is the pharynx is kind of like in the middle here, and then there is this cavity around the pharynx called a pharyngeal cavity, and then the gut pocket kind of has like a triple arm arrangement like this. So here's the gut pocket, something like this. Okay, so that's kind of how the gastrovascular cavity is arranged in the organ. All right, so if I were to do like a cross section through this, like let's go ahead and take a look at a cross section through the anterior end. This is called the anterior cross section. We have a pharyngeal cross section. CS means cross section. And then we have a posterior cross section. So I have a slide here that has a whole mount of the planaria, the flatworm. So you want to look at the whole mount one. And then I have another slide that has the 3 representative cross sections of the body here. So let's go ahead and draw what the cross section looks like. The anterior one looks like this. I'll do them in order. The anterior one is kind of like this. It has the body flattened like so. And this is ectoderm. It's a single cell layer of outer tissue. The gut on the inside is usually kind of folded, so you'll see like something like this. And sometimes there's little side pockets of the gut like this. And the gut is made of endoderm. So the gut pocket is endodermally lined. But all of this internal stuff in here, which is mostly muscle and connective tissue and internal organs, this is all what's called the mesoderm. Gesundheit. So all of this red stuff here is the mesoderm, and it's a combination of muscles, internal organs, connective tissue, gonads, etc. So basically the body is filled with only a single cell layer of ectoderm, only a single cell layer of endoderm, but it's multiple cell layers of mesoderm. This is why the mesoderm kind of controls the body. So this is our anterior cross section here. Looks something like this. The pharyngeal cross section is one in the middle, looks something like this. It's usually a larger section. Okay, there's our ectoderm. I'm not going to label all the dermal layers, they're the same color. Okay, so you can kind of get that. So in the pharyngeal cross section, we have the pharynx is lined with endoderm on the inside, but it has actually something like this going on. You'll see in the cross section what looks like a donut inside there. Come back to that with mesoderm. And then we have usually some side branches here, like this. Like so. So anyway, so what we have is the endoderm's in blue. It's the lining of the pharynx inside and the gastrovascular cavity on the lateral side. You can see the lateral side has the gastrovascular cavity there. And then we have all of our muscles here and internal organs and mesoderm. Basically, mesoderm fills all the rest of the spaces here. The dot is kind of representing tissue. And then there's muscle basically ringing the inside of the pharynx here as well. So there's like the pharyngeal cross section here. And what you can see here is the pharynx is suspended in this little cavity here. This little cavity here is called the pharyngeal cavity. Okay.
I took pharynx and just put
this
pharyngeal.
pocket here is the inner lining of the pharynx. And then this is the gastrovascular cavity. Oops. Okay, so that's what you're going to see in the pharyngeal cross section. All right, the last one is our posterior cross section. And you can kind of predict what's going to happen here. We're going to have lateral gastrovascular cavity, there's going to be no central one. So this one, it's kind of like that, but no central one here. And that's how you can recognize the posterior cross section. Something like this. We have our ectoderm. We have our endoderm is only on the lateral sides. This. And there's no central gastrovascular cavity. So we have our muscles, organs, like so. There you go. So this would be the posterior cross section. So on that the point there is we're looking at endoderm, mesoderm, and ectoderm, and we're looking at bilateral symmetry. And so when we're looking at our planaria, here is the diagram that you see. I have slides that are whole mount slides, and what they did is as their last meal, they fed them some stained food, and the food stains the gastrovascular cavity, so you can see how branched it is. Here is what the three representative cross sections kind of look like. The anterior one basically has a central gastrovascular cavity pocket, and maybe some side branches, but there's basically gastrovascular cavity in the middle, and some side branches. The pharyngeal one, you can clearly see, so it looks like a bullseye. You can see there's the pharynx, there's the inner lining of the pharynx, here's the outer lining of the pharynx, and then there is all the mesoderm there, and it's in this pocket that is called the pharyngeal cavity. The pharyngeal cavity is lined with ectoderm. This is, the brown is ectoderm, ectodermal line. Hint, hint, that's a good test question. So this pharyngeal cavity is lined externally with ectoderm, even though it's inside the body. And then we have lateral gastrovascular cavity pockets. And then the posterior cross section has lateral gastrovascular cavity, but nothing in the middle. Okay. So I'll have a slide on the test, and I'll have you probably you'll have to tell me what region of the body it is, anterior, pharyngeal, or posterior. Hint, hint, it's probably going to be pharyngeal, right? Just saying. Right? So, how much easier can it be? All right, let's see who gets it right. Here is how the flatworm feeds. Is they use this little pharyngeal tube to they're scavengers. They'll feed on decomposing things, organic material. I actually have some live ones here. If you want to mount one of these, you can I have these depression slides. These depression slides here are slides that have like a little cavity in them, like a little dip in them. So you can take one of those little flatworms, put it into a depression slide, put a cover slip on it. Your big microscopes, the Congo microscope, you're going to basically be focusing in on like the head or whatever, they'll crawl around. But we have these dissecting scopes, so you can take a look at them on the dissecting scope. But they're really kind of neat. So you can try those out. If you use these depression slides, make sure you clean them off and put them back over here. They're special slides. Okay, so that's kind of the basic idea of what the flatworm is. We have a new thing where we have triploblastic. Everything else we see this semester, including you, is triploblastic. You have 3 tissue layers. Everything else we see this semester will have bilateral symmetry. So it's kind of like a common theme for the rest of the animals that we have. Okay, let's go ahead and take a look at the classification. We have 4 classes. I only have 3 here to show you, but we have 4 total that you might see around nature. Class Turbellaria. Class Turbellaria. Class Monogenea. I don't have any Monogenea, but you might see them if you go fishing. So I'll show you that one, but I don't have any specimens of it. We have the class Trematoda. And then we have the class Cestum. Okay, so that's our 4 classes. Again, the Molten Star won't be on the test. We don't have any, but I want to show it to you because you've probably seen it big. Okay, so the Turbellaria are the free-living flatworms, commonly called planaria. 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 I have a slide of planarians. You want to look at the whole mount and you want to look at the cross section. For example, the whole mount looks like this, and the cross section would look like this. Okay, look pretty good. So basically, that's what we're looking at in a planaria. Got it? Pretty common. They live in ponds and creeks or whatever. Some people confuse them for leeches, but they're not leeches. There's also, if you have an opportunity when it starts raining eventually, there is another free-living flatworm that occurs in our lawn here or in gardens in the Reading area. And that's this one on the right. It's called bipalium, or a hammerhead worm, because its head has this big kind of spade appearance here. This is the head region of it. It has kind of like a spade head or hammerhead. These can be quite long. I mean, they're about 10 inches long, sometimes 8, 10 inches long. They are earthworm vampires. They're spooky. When you see them crawling around, you think it's like a big earthworm, but it looks different, not segmented. Anyway, the hammerhead is full of sensors, and they can smell or taste earthworm slime as they leave behind a trail in the dirt or soil. And when they finally come upon an earthworm, they will use their pharynx to eat a hole in the earthworm and suck all the blood out. So they're basically little earthworm vampires. People who have worm farms or raise earthworms for worm soil, etc., hate these things because they'll annihilate your worm population. These are introduced species. They came from probably Southeast Asia, like that area, probably with ornamental plants. They came from the soil, and they're now invasive all over the U.S. Supposedly, these have a toxin, tetrodotoxin, which is the same toxin that's found in pufferfish. So don't eat them if you ever see one. Not good. But anyway, supposedly, like if the dog eats them, they can get poisoned. I guess supposedly it's even in this, if you handle it with your hands, like there's a species in the south that if you handle it, your hand will get numb because of the toxin. So anyway, interesting work. If you see them, bring them in. They're really spooky, but they're pretty safe. They're not going to hurt you. Otherwise, in the ocean, free-living flatworms are pretty common. These are called marine planarians. So these are marine, freshwater, and then this hammerhead worm is kind of semi-terrestrial with wet soils. Okay. I don't have any specimens of these, but I do have specimens of these. So plan on a turbolaria or two being tested. Okay, monogenea. I don't have any specimens of it, but the common place where you find these is on they're all fish ectoparasites. So if you go fishing and you see little white spots either on the fish's body, or usually where you find them in freshwater is on their gills or on their pectoral fins. Let me go ahead and draw a fish here. Let's see here, they draw a fish. So if you go fishing like blue, I see them on bluegill a lot. They look like a little bluegill here. Anyway, where you find them is either hanging off of the gills like this, or attached to the back of the pectoral fin. So you'll find them, as ectoparasites on fishes. And what they have basically is they're called monogenea because they have one oral sucker. So the body looks like this, and they have one oral sucker here. They also have this really interesting, weird, like clasping structure here. Usually, it's kind of like a ring, like a disc, but it's called the epistosaur or episaptor. And it's like a kind of a Velcro hook on their rear end. So kind of interesting. I don't have any specimens, so none of this is going to be asked of you on the test. But if you go fishing, just take a look, especially for bluegill or crappie. You find them kind of attached to their fins and the gills. Or if you go ocean fishing and you get scolded, you find them on the bottom. They can't hurt you, but they're all ectoparasites of fish. Okay. Sound good? Trematoda, this is where we'll spend a lot of our time here. The trematoda are all internal parasites, or endoparasites of vertebrates and snails. They actually have usually multiple hosts. So a couple of things about these is they have oral plus a ventral suckers. They have two suckers, an oral sucker and a ventral sucker. And these are attachment suckers they can use to attach their host. Also, all of these trematodes, for some reason, have an intermediate host of a snail. So there is a snail, usually a freshwater snail, intermediate host. And what that means is they're obligated to, at some point in their life cycle, to occupy a snail to survive. So they have to get to a snail to have one stage in the life cycle, the superior stage, to be produced. Otherwise, they have a vertebrate as their main host. Okay? So intermediate host is a flying snail. All right. The first one we're going to look at is Clonorchus. There's three of them to look at here. These are all human parasites we're talking about. Clonorchus, Clonorchus fasciola. Fasciola and schistosoma. Okay, so there's the three specimens I have. I have Clonorchus here, I have fasciola here, and I have schistosoma there. All right. Clonorchus is what's called the human liver fluke or the Asian liver fluke. And what I want you to know, I'm providing the Center for Disease Control life cycle posting on this. What I'd like you to know is something about the anatomy of the organism's kind of body parts. But also I'd like you to know about what the life cycle is in terms of the hosts. So how do you get this parasite? This is a fairly common parasite like in Southeast Asia, Central Asia. And it's caused by eating raw fish. So eating raw fish is kind of the primary way you get this disease. So I'll do Clonorchus over here. This. So Clonorchus looks something like this. About maybe a couple centimeters long. And the key features you want to find are, here we have an oral sucker. And here we have a ventral sucker. The oral sucker there is the mouth. And the ventral sucker is where the gonads, the testes and ovaries exit the body there. On this diagram, these letter labels are what they're talking about. OS is oral sucker, PH is pharynx, VS is ventral sucker. You get the idea. I'll mention the other ones as well. Okay, for the gastrovascular cavity on this one, it's a little bit abbreviated. It's got a pharynx here, and then it has like two lateral tubes here, like down the side of the body, something like this. So, what you're seeing here is the, I think the CE here is referring to the gastrovascular cavity. I'm not sure what CE stands for. But basically this is the gastrovascular cavity, GV cavity. Okay? Now these are hermaphroditic, meaning they have both male and female parts in them. And you know, how you can tell male versus female parts is kind of what's in them. The female structures, which include the ovaries, eggs, oviduct, and uterus. So it's kind of a combination of all these features. I'm not going to ask you to differentiate them. But what you can usually see is kind of in the middle of the organism they have this kind of what looks like I don't know, looks kind of like spaghetti or looks like noodles in there. And specifically, how you can tell is you can see there's eggs in this region. So this region here with the eggs in it would be the female structure. And the female structures include the eggs, the uterus, ovaries, etc. So the female structures are kind of in the middle. The male structures are kind of in the posterior regions of these. If I can use a different color here, here I'll use green or light lime green. The male structures just kind of look like pom-poms, like kind of like a bushy pom-pom. And this is the male testes. So if you think about pom-poms, you can think of like gonads, right? There you go, don't forget that. So the male structures, the testes, kind of have that pom-pom look, and that's because they're kind of fibrous and producing sperm. And then what happens is the testes and ovaries, basically the vas deferens and ovaries, etc., will all end up at the ventral sucker, and that's where the fertilized eggs exit, and that's where the sperm can exit. They can self-fertilize, and they can also cross-fertilize. So that's the clonorchus there, and there's some right there on that thing. Any of that third name, I think that's the male versus female structures. Gastrovascular cavity, oral and ventral suckers, etc. Okay. The life cycle is such: humans get the parasite by eating raw, uncooked fish. Fish that has been contaminated and is raw, and then they get it. And then the parasite basically migrates to your liver. It's a liver parasite. And then the parasite produces eggs, and the eggs are circulated back into your bloodstream, back to your digestion, and the eggs are passed in the feces. So when you go to the bathroom near a pond or near a waterway, the eggs end up back in the water. The eggs are eaten by an aquatic snail. And in the aquatic snail, you have the several life stages that occur. We'll talk about that with fasciola. But out of the aquatic snail comes this thing called a cercaria stage. Cercaria is like this, it drills into the next host. Cercaria typically is like a ectoparasite in terms of it attaches and it drills into the tissues. So they drill into the meat or the muscle of a fish, and then the human eats the fish. And so that's how the life cycle kind of goes. The way to control this parasite is if you can kill the snails in a pond or waterway, you can control these parasites, and that's how treatment codes are controlled, is by getting rid of the snail population. Usually with like sulfur compound. Copper sulfate is a great way to kill aquatic snails. Okay, on to the next one. Fasciola, the body of fasciola kind of looks the same, so I'm not gonna draw fasciola. It's a bigger worm. They're about the size of your thumb. These fasciola parasites. They're called a sheep liver fluke, although they can also infect humans as well. And they're pretty big, like you can see on the left there. It's like a couple inches, an inch and a half, almost 2 inches long. This one for the fasciola, I want to focus in on the life cycle. So here is how fasciola kind of operates here. Fasciola is called the sheep liver fluke. So I'm going to do my best to draw a sheep here. I draw invertebrates way better than I draw vertebrates here. Here's my sheep. So if you're in doubt if this is a sheep, not a cloud with sticks on it. What the heck? They're all right sheep on there. That's like, yeah, I know more good. Or like a fluffy rat. Okay, so in the sheep we have these parasitize the liver. So they're called the sheep liver fluke. These can also occur in deer if you're a deer hunter. And if you're eating deer livers or whatever, sometimes when you slice the liver open, you can see cysts in the deer liver. They occur in cattle also. Anyway, the fluke lives in cysts in the liver. Okay, so this is the what's called the liver fluke, and this is the adult stage. So what happens is the sheep or cow or human or whatever poops out, or you defecate out the eggs that are produced by the liver flukes. The eggs migrate back into the circulatory system and into your intestine. And ultimately what happens is the eggs are deposited out the rear end. The eggs will be taken up or consumed by an aquatic snail. So I mentioned that snails are a key host of these. So this is a snail. Can't drop that earth. So the snail basically ingests the eggs or the larval stage. And in the snail, you have a couple stages. The eggs turn into what's called the miracidium. And the miracidium stage looks kind of like a little paramecium. Remember last time we were looking at the larval stage of the Aurelia, Aurelia planula? It kind of looked like a paramecium. This kind of looks the same way. And in fact, if I were to have those on a test and not labeled, you probably wouldn't be able to differentiate them. But the miracidium is the stage that initially infects the snail. And inside the miracidium are these cercaria. So inside of the miracidium, cercaria develop, and they're asexually produced. And the next stage is the stage called the cercaria. And the cercaria kind of look like sperm cells or tadpoles. So these are the cercaria. Cercaria, C-E-R-C-A-R-I-A. It's on the page, so you can look at your document there. Okay, these are the stages that emerge from the snail and then go on and infect the next host. The next host is grass. It is a vegetation. So aquatic grasses. This is grass stems or vegetation, and the cercaria will invade the grass and insist inside the grass. And this is called the metacercaria. Meta means after or behind. So, so the metacercaria is embedded in the grass stem, and then obviously the sheep, when the wetlands are drained or when the rice stems are drained, for example, the sheep will graze on the grass and get reinfected. And so this is kind of the life cycle here of the fasciola. This is fasciola. And how humans get it is eating contaminated vegetation, right? If you eat a salad that has this veg in it, and you get the metacercaria, you can get infected by this. So don't eat grass, right? Or if you do, make sure it's cooked really well. There you go. Question? Okay, so I have a whole life cycle here. So the adult liver fluke is the big one, and then the other tray has the miracidium, cercaria, and metacercaria stage. I think if I were to ask you about any of the stages of that species, it'll probably be the cercaria stage. That's the most interesting one. I'll tell you why. Because the next one we have is called schistosoma. And schistosoma causes a disease in humans called schistosomiasis. Schistosomiasis is a blood fluke. This is a fluke that lives in your bloodstream or blood systems. The schistosomiasis specifically is, I think it's a disease in mostly Africa, and maybe it occurs in South America as well, but I think it's mostly Africa. Anyway, this is a case where if you go wading or swimming in a pond that's infected with this, you can get schistosomiasis. And what happens is you go wading and swimming, and the cercaria will actually drill into your skin. Here's an example of someone who's been infected with cercaria of schistosoma. And normally what happens, like in Africa, it's people who are wading, either washing clothing, or they're washing food, or going to the bathroom in the creek or water or whatever. And normally what happens is they drill into your feet. And so how you can tell that you have it, or you got infected, is you have basically like pock marks or like a rash that develops where the cercaria forms. This causes a blood parasite. I have some slides of these. This is actually a male and a female together. They're hanging out in your bloodstream, attached together and reproducing. I believe the male is the small one, the skinny one, and the female is the larger one. But that doesn't matter. You should just know that when you're looking at the schistosoma on the slide, there's actually both a male and a female there together. That's why you see like two heads and two tails. It's a combination of male and female. Male wraps around the female. Okay, so then what happens is they produce eggs. The eggs that end up in your intestine. You go to the bathroom, you poop by the pond, and the eggs get back into the waterway, and always, there is a snail as the intermediate host. The snail is where the myristidium develop, and then you can see these little white spots in this cup. Those are all cercaria. And then here's cercaria underneath a microscope. You can see they look like little tadpoles or sperm cells, little darts. Okay, so that's schistosoma, which causes schistosomiasis. There is a related species that occurs in California called swimmer's itch. And swimmer's itch is what's called cercarial dermatitis. This is a schistosoma parasite that is intending to infect waterfowl like ducks and geese. So where you find swimmer's itch is where you find basically flocks of ducks and geese will basically settle into a pond or migrate. Ponds that have ducks and geese. And those ponds usually have snails, will oftentimes have swimmer's itch. And what happens is people will go swimming. Lake Pillsbury in Mendocino County is like notorious for people getting swimmer's itch. I actually got this one time. I was swimming in a lake in Santa Rosa. I used to go to mountain biking, and I didn't think about it. And within a day or two, I had a rash that basically probably was swimmer's itch. So what happens is the cercaria are intending to drill into the duck or the goose and complete its life cycle like this. But accidentally, the cercaria drill into your skin, and because you're not the right host and your skin's too thick, the cercaria dies about 1 millimeter under your skin. And then your immune system attacks it, causing the rash. And so the swimmer's itch, cercarial dermatitis, is basically schistosoma that was supposed to be a species that infects ducks and geese, but accidentally infects you and causes a skin rash there. There you go. Fun stuff, huh? Okay, so I have a slide of schistosoma. Make sure you check out those. A good question is the specimen male or female? And it's actually both, right? It's male and female. I think the slides say incopulate, which is okay, so class Cestota. The Cestota includes the tapeworms. This is the tapeworm. These are bizarre. It's almost like they're from another planet. They're totally bizarre. A unique feature of tapeworms is they don't have a gut tract at all. There is no gastrovascular cavity in these. They've lost them. What the tapeworm really represents is it's like a colonial flatworm. And you've heard this colonial theme before, right? Like the Portuguese man-o-war was a colonial pollen or a hydroid colony. We've seen colonial protists, right? So anyway, the sponge, is kind of like a colonial aggregation of cells. So this is like a colonial flatworm. And there are multiple worms like attached together, and they mature through a specific life stage. I'm going to go ahead and draw a tapeworm here, and I'll draw the three main regions that you need to know. There's like three main parts. The head of the tapeworm, so this is the cestota. Last cestota. Tapeworm. Okay. The head of the tapeworm is called the scolex. And it has like usually a couple suckers and hooks. So the scolex has hooks and suckers attached to what's called the rostellum. I'm not going to ask you the term rostellum, but basically the scolex is the head region. What happens is how you mostly get tapeworms is you ingest the eggs accidentally. So you either with your food or by, sucking on your fingers after you've been digging in the soil or whatever. So you get the tapeworm by ingesting the eggs. The eggs hatch in your stomach. The scolex basically is the first worm that hatches out of the egg. And because it has these hooks and suckers, they attach or embed themselves in your small intestine. So they kind of hook into your intestinal lining and live in your small intestine. Once the scolex has attached, they begin producing what are called proglottids. The name proglottid means before the tongue, right? So these are the pieces that end up getting ingested. So these proglottids that are produced right behind the scolex, it's kind of like a Pez dispenser, right? What happens is the scolex asexually produces these. So this is one that's just been made, and when it's made, it pushes the next one back. So as you go back, they get older and older and older. So these near the scolex end are what are called immature proglottids. The immature proglottids you can recognize because they only have male structures. So in the immature proglottids, you're going to see what looks like developing, they're developing testes, but again, they kind of look like little pom-poms, little gnats, right? So the immature proglottids basically are the male's testes forming. Usually the ones that are just produced, you can't see anything in them. But as you work your way back, you'll see they become kind of male oriented. You'll see kind of some stuff developing in the more anterior ones. But as they kind of go to the end, they're essentially becoming male producing testes. Okay, the dashed lines here represent 20 or 30 segments down the way. What you're going to find are proglottids that are more developed, and they're kind of become more like rectangular. And these are what we call mature proglottids. Okay, these mature proglottids, in the anterior ones, the testes may still be present, but the testes begin to kind of dissolve and go away. And by the time you get to near about 2/3 the way down the worm, the testes are gone. But what takes over, and the theme here is in mature proglottids, you begin to have developing ovaries. So the female structures begin to kind of develop, and occur in the mature proglottids. So the mature proglottids, I get it. It's a progression, right? You have nothing here. You have testes forming. You have testes and ovaries beginning to form. The testes disappear and the ovaries kind of take over, right? So it's like a progression, right? It's not like you have suddenly testes gone and ovaries just appear. It's like the testes develop, the testes dissolve and go away. The ovaries develop, there you go, right? So basically we have the female ovaries and oviducts are forming here in the mature proglottids, in the middle region. Okay, and then finally, at the end of the worm, we have what are called ripe proglottids. And these ripe proglottids are ripe because they have eggs in them. So what you're going to see here is you're going to see the ovaries still, but the ovaries will be loaded with eggs. And as you go to the very end, the eggs become more prolific. And these have eggs, as well as ovaries. And these break off one by one at the end. And when they break off, they get passed out with the stool, feces. And that's how they're passed out, to be re-ingested. They break off the end as like basically a packet of eggs. And if your dog or cat has tapeworm, you can usually tell, because when the dog poops, right away, take a look at it, and you'll see little white worms crawling on the poop. What happens is the worms, loaded with eggs, will migrate underneath the poop. The poop begins to decompose, and in the case of a dog tapeworm, which is specifically dog tapeworm, it's Diphylidium caninum. This is dog-specific tapeworm, which can also infect cats or infect humans. What happens is the worm is ingested by a flea larva. So normally in like a coyote den or a fox den or a wild dog's den, you would have like the dogs there, the puppies there, and everyone's kind of pooping around the den or pooping in the den. And what happens is the tapeworm proglottids, ripe proglottids, will basically migrate underneath the poop. Flea larvae are scavengers, or they eat decomposing materials. Flea larvae will ingest the tapeworm eggs, and then the tapeworm larvae will develop in the flea. The flea then matures into a mature flea and jumps on the dog and basically bites the dog. Fleas are blood parasites, right? So how the dog gets reinfected is not by the flea bite. This is a test question. How the dog gets reinfected is when your dog has fleas, what do? They chew and they lick. But basically the dog will chew and lick the fleas, and the dog swallows the flea. So usually when a dog is licking or grooming themselves trying to clean fleas off, they'll essentially ingest or swallow the flea. And that's how the dog gets reinfected by tapeworm. And so anyway, humans can get this by swallowing a flea. And how would you swallow a flea? By accident, right? So it's pretty rare to get dog tapeworm in a human, but it is known to happen. Cats can get dog tapeworm also, but it's not as common as tapeworm. So you can see tapeworm in cats as well. It's pretty easy to treat. You just go to your vet and ask for, they give you 1 pill or 2 pills, a follow-up pill, and it cleans them out within about 5 days. Or I think you can get the pills at like Tractor Supply or like a pet supply places. Pretty easy. Questions? Okay. Cork tapeworm is the more serious parasite in humans You get pork tapeworm by either eating contaminated pork meat that's not cooked, or worse yet, you get it by eating poop. So if you inadvertently ingest pork or livestock feces, basically, if a vegetable garden has been fertilized with manure that hasn't been properly composted, you can get tapeworm eggs on your vegetables, and when you're eating your salad, you get infected with tapeworm. And so you could basically get the same kind of life cycle here, but you're basically ingesting it either through uncooked meat or through accidentally ingesting the eggs and feces. The problem with pork tapeworm is one way the life cycle can go is the tapeworm ends up in your intestine and you have tapeworm. It's great, you lose weight, you're never alone, right? You have always everybody. The other way that's a problem though is if the tapeworm ends up in the wrong location. So if the tapeworm ends up in the wrong location, they can end up in your brain. And this is an MRI scan of someone's brain that has tapeworm cysts in their brain. And it's called cysticercosis. It's a tapeworm cyst that form in your brain. And the problem is you can get infected by these. Usually what happens is I knew someone who died of this. At Berkeley, there was a postdoc there that died of this while I was there. And what had happened was he was living in Thailand a decade before. And presumably what happened was he was eating vegetables like a salad or whatever that had been fertilized with pig manure or human manure, either way. And he'd ingested tapeworm eggs through his salad or food, contaminated food. And then the tapeworm larvae migrated to his brain. But he didn't start having seizures or neurological problems for about 10 years. And didn't know what was going on. He thought he had epilepsy or something. When he went to the doctor and they did an MRI scan, they found something like this. They found little pockets or cysts, kind of the size of like a pea each in his brain, dozens of them. And once you get to that stage, it's terminal. You can't fix it. So he ended up dying of, basically his brain gave out on him. So that's the issue with the tapeworm. If you can get the pork tapeworm specifically, it can migrate to the wrong organ and cause this cysticercosis. I have dog and pork tapeworm here. It's Dipylidium caninum is the dog tapeworm. Tania is the pork tapeworm. I'm not going to ask you to differentiate the two. What I do want you to know, though, is if I have a scolex on there, the questions would be the scolex and the immature protolatids together. You can see those both in the same slide. The immature protolatids have developing testes. The scolex has basically the attachment part of the worm. Because these worms are so long, these are sheep tapeworms. These worms are like, a couple feet long. The ones that infect Tania, the ones that infect humans, can be upwards of 6 to 8 feet long. So you can't put them all on one slide. So other slides will have mature and ripe protolatids on them. And the mature protolatids specifically have developing ovaries. Even though the testes might still remain, the main theme with mature is that ovaries are developing, right? Got it? I'm saying it that way. And then the ripe protolatids, the ovaries are still there, but the eggs are formed. And again, this is a progression. It's kind of like it goes progressively through the stages. So you'll have nothing, you'll have testes, you'll have testes and ovaries, you'll have just ovaries, you'll have ovaries and eggs, you'll have just eggs. So it's kind of like a life cycle as it goes down to matures, right? So on the test, I'd like you to know what's contained inside the immature versus mature versus ripe. Got it? Not too bad. I've laughed for an hour, as usual. And we have 1, 2, 3, maybe 4, 5, 6, 7, 8 slides to look at today. So that's not too bad. And again, I'm kind of telling you along the way what I'll probably have on there. Certainly the pharyngeal cross-section of the planaria. Probably a cercaria of the fasciola will be on there. Certainly one or two tapeworm slides, scolex and immature and mature and ripe separately. What else? I think that might be it. So you can imagine like 4 or 5 stations that have something to do with flatworms. All right, there you go. You have the rest of the time to finish that out yourself and get your microscope. I'm going to take this away here.
I don't like this. Is it kind of like the same? Oh, yes. Yes. Okay, so I'm gonna